Yes, good evening to everyone. Welcome to day 10 session on QSAR and Pharmacophore modeling. So before proceeding on today's topics, today is the last date for this drug design and molecular mechanics. From tomorrow onwards, we'll be discussing to a new chapter called vaccine design. Okay, so today I'll be completing molecular mechanics and drug design together. That is, first I'll be discussing on QSR, that is a ligand-based drug design. So before going to today's session topics, can we recap the last session topics? I show you what exactly did we discussed. We discussed MD simulation and we have discussed molecular docking analysis. Can I ask you a few questions related to this? First, related to MD simulation. Okay, so mention few examples of MD simulation softwares. So we have seen few softwares where it can do the MD simulation, isn't it? So what are that softwares? And I showed you few URLs also. You remember few URLs and explained you even in the tutorial how to do the simulation. Yeah, come fast with the answers. Don't restrict up to answering the poll alone. Even these kind of descriptive answers you can try to answer. Examples of MD simulation softwares. No one? Try something. At least one software. If not, whatever you remember. No? It's very bad. Actually, we have discussed so much things. Okay, so nearly 1 hour. I have extended more than an hour. If you see the recorded video, it comes more than 1 hour. But not even a single participant is interacting with the MD simulation software. So, if you see the MD simulation softwares, we have discussed this: Abalone, Amber, Charm, Desmond, Chromax, Chromos, NAMD, Q, Yasara. Other than this, we also discuss something that Discovery Studio also has. The Discovery Studio is a package, it has its own kind of a simulation software. Similarly, in Schrödinger package, the simulation software to be worked out is Desmond. And I have taken you to this tutorial, like the ligand receptor interaction tutorial, and I showed you nearly 8 steps to undergo the simulation. To perform MD simulation, we have to follow sub steps in that 8 major steps of simulation. Okay, so we have discussed, isn't it? So just recall everything because this is important, of course. Very good. One is answered. Chromax and Amber. Good. Now let me go ahead with the today's session topics. So as mentioned, we'll be discussing initially on ligand-based drug design. In ligand-based drug design, previously whatever we discussed, molecular docking, MD simulation, this comes under structure-based drug design. It means that we should have 3D structure of ligand as well as 3D structure of receptor to understand the binding interaction as well as the stability. Okay, we need the 3D structure of these two molecules. But whereas in the ligand-based drug design, we need just the 3D structure of ligand alone. Even if you don't have a 3D structure, if you have a 1D structure, what is 1D structure of a ligand? I told you this will be in the form of Smiles format, canonical Smiles format. The expansion of Smiles is small molecule input line entry system. This is the expansion of 1D format of a chemical compound or a drug. Okay, so if you have 1D, even it can convert it as 3D. Okay, so there are plenty of tools available. And if you see, the first thing what we discuss is QSAR. Here, Q stands for quantitative. Okay, so defaultly, if you mention Q, it is quantitative, not the qualitative. If you have, if you are discussing on QSAR, there is also one thing called SAR. QSAR is different, SAR is different. If we find this SAR, it default it means that it is a qualitative approach. So only this is qualitative. That is, the quantity is not required. For example, activity, if you determine or if you determine the concentration of the concentration of the drug is determined only in this quantitative approach. In qualitative, we don't need the concentration. We need just whether the concentration is low, medium, or high, something like that. Only we represent these kind of terms. But for us, we can also measure here with values. For example, 120.3 micromolar, something like that. Okay, the quantity is measured. And ACR stand for structure activity relationship. Now let me take you to the further thing. We will be discussing on statistical parameters and QSAR modeling, types of molecular descriptors, and a tool for QSAR, ChemDes, and few more software examples, types of QSAR, and then we discuss about pharmacophore modeling and futures of pharmacophore and pharmacophore mapping, especially form mapper. That is a freeware to practice. Now about QSTR. As I mentioned you, the expansion is quantitative structure activity relationship models. So these are generally regression model or even a classification models. Generally, it is used in chemical and biological sciences and engineering. And if you see this QSAR, okay, this regression model uses two variables. Predictor variable, also called as dependent variable, and the another thing is response variable, which is an independent variable. Okay, for example, if you have a graph, okay, like for example, if you have molecular weight of nearly some 20 compounds in a data set, and if you have log p, okay, that's what you can see even in zinc 15 or zinc 20 database. You remember, we can able to uh, uh, download the set of molecules through tranches in zinc 20 database. So where it is mapped, the molecules are mapped with molecular weight versus log p. So for example, if you are having these two properties for 20 molecules, okay, so 20 molecules, something like that, I am showing in the different dots. There are 20 molecules here, something like this. And if you perform the regression method, in the regression method, we see that all these dots are mostly joined. Okay, we draw a straight line which can join much of the points. Then whatever line, linear fit we have drawn, so this is actually measured through y equals mx plus c. So y is a independent variable, x is a dependent variable, and m is the slope, and c is the intercept. So whatever method we use now at present is linear regression model. So in the linear regression model, we can able to predict the y and x values. I mean, we can able to predict the y and x values from this method. So like linear regression, we have different regression methods: polynomial regression, multiple regression. So like this, we have log regression. We have different types of regression methods. And one more thing they mentioned is classification. Under classification, okay, so if you can able to draw for example, if you have same thing like molecular weight, instead of molecular weight, we have something else which can distinguish even like a lipophilicity. Okay, lipophilicity in y-axis, and something like log p in x-axis. So if you have like this, okay, and if I have 20 drugs, and these 20 drugs, okay, if you find like this, and we also have few more drugs like which I show in different color. Now I take a different color. If I have drugs like this, okay, then according to the classification method, okay, according to the classification method, so we can draw a line and we can segregate this versus this. These blue color drugs are now distinguished with the violet color drugs. How is it possible? Through drawing one kind of a curve, okay, which can clearly separates drug 1 to drug 2. This blue color can be a more lipophilic in nature, and whatever you find this violet color might be a more hydrophilic in nature. So if you want to classify the drugs, okay, whatever you find in Swiss ADME, if you see in Swiss ADME, to distinguish lipophilic drugs from the hydrophilic drugs, we have one diagram called boiled egg diagram. You remember that? That is going to classify like in a egg, okay? We have a yolk. If any drugs, okay, falling in the yolk region, then it means that it is a highly lipophilic drug. Instead of that, if drug, whatever we are searching, okay, is lying somewhere in the white albumin region, it is hydrophilic drug, isn't it? So this is also using a classification method to distinguish one type of drug with another drug. So this is all. This is what I mean classification model. Decoration is this, and classification is this. Got it? Decoration, classification. Now let me take you further. So if you want to link the variables, okay, through the yx kind of equations, obviously this kind of equations we generally use it for calculation of activity. Okay, for further information on we can see in the subsequent slides. Before going to much more information about QSAR, first we can discuss on the history and development of QSAR. The QSAR term, came from these related things. Actually, exactly if you see the QSAR was proposed by Hans in 1964. You know the origin of Unix, C++, C, etc. So this comes around 1960s. In 1959, 1962, we can find the origin of unique C languages, etc. So during that time itself, they come up with this kind of QSAR approach. But before that itself, it already started. Okay, like if you see the Fraser has come up with a kind of a function. Phi is a function of concentration. You know, this is whatever I am telling the phi is equivalent to y, and whatever function of x, that's what we give like mx plus c. Okay, such a kind of a linear regression to predict biological activity. Generally the biological activity is what we predict from this y variable. Whatever they shown phi is the activity. Okay, generally if you see, if you want to determine activity, antimicrobial activity for a compound, the antimicrobial activity of a compound is generally determined as negative logarithm of MIC 50. MIC, that is minimum inhibitory concentration 50. You know how the antimicrobial activity is checked? Initially, we take the agar, we pour the agar in the Petri plates. Later, we place the antibiotic disc, isn't it? So based on the zone of inhibition, we can able to measure. So whatever I mentioned like zone of inhibition, that is if you measure minimum inhibitory concentration of 50, then whatever you take negative logarithm of this, that's what we predict as activity for that antimicrobial drug or compound. Similarly, we can able to determine as many number of compounds. If you have 20 compounds, for 20 compounds, you determine activity. And whatever we predict activity for the 20 compounds, we call that as training data set. The training data set is a large population data set. You know, here the variables are predicted. How from a dependent variable and independent variable is predicted. Like this, you can able to see all the variables. I mean, initially we already predict the activity from the wet lab experiments, and after predicting the activity, if you want to find for one sample for which the activity is not known, that we keep a term, we use a terminology like a test data set or test data. If it is only one data, it is test data. But if you have 2 to 3 compounds and for which the activity is not known, if you want to break the activity of the 3 compounds from the training data set of 20 compounds, then only we are using this kind of equation, linear regression equation. From linear regression equation or any other regression equations, we can able to determine the activity. Okay. So one thing we should remember in QSAR that we have 2 important terminologies. One is training data set, which is of large population, and test data set of small population. Okay, so more about this, if we discuss in subsequent slides, we can come to know about that. If you see this in 1900, from Mayer and Orton, they come up with the lipoid theory of narcosis. Like if you want to distinguish the lipophilic drug and hydrophilic drug, these kind of distinguish was done by the Mayer and Orton. And Hammett, they come up with the electronic sigma constants. Okay, you know that we have electrostatic interactions, van der Waals interactions. These comes under non-bonded interactions. Of course, weak interactions when compared to bonded interactions. If we come across electrostatic kind of interactions, we can able to find these kind of electronic sigma, like whatever the instrument measures, the electrostatic parameters, this can be measured. And actually, for this, Hammett has proposed this in 1930. And in 1984, from Andrews, he come up with affinity contributions of functional groups. And good for, the Kramer come up with 3D QSAR. If Hans come up with a QSAR term in 1964, in 1988, the 3D QSAR was proposed by Kramer. And whatever we discussed initially about Lipinski rule of 5, okay, this started somewhere here itself, 1997. Okay, Lipinski rule of 5. You remember? 3 out of 4 rules, even if it satisfies, then we call it as a compound behaves like a drug. So this was proposed by Lipinski at 1997. Of course, these are all the futures of QSCR, and this is the Eastern development of QSCR. So the origin of QSCR was developed more than 50 years ago. One thing you have to remember, like however I mentioned, the activity that is antimicrobial activity of a compound or drug, similarly, we can able to determine activity of any biological activity, okay, through QSAR. For example, if you want to measure the toxicity of a drug in small animals like mice or rats, generally we try to determine ED50, effective dose concentration 50. Like for microbial, we use MIC50. But for testing the dosage patterns, especially the toxicity of in animals, we measure through effective dose 50 concentration. So if you want to determine the activity for that, okay, defaultly this is given through this kind of a relation. Negative logarithm of ED50. Might, if it has some kind of a bias, we can link with some other constants. We can link with some constants. Got it? Generally, we use this is a simple understanding how the activity is determined from the effective dose concentration. Similarly, we can find other parameters of can also be used to determine activity. The activity, whatever I mentioned, is a correlation of physical and chemical properties of the drug. Okay, so it can be of any properties like even pKa value, inhibitory constant values. Even through this also we can use a correlation to determine the activity. And generally, the binary terms in qualitative structure analysis, if it is a quantitative, we measure these through values. There are some specific values, range of values. But in qualitative structure activity relationship, that is what we call it as SAR. For SAR, we simply mention whether that compound is active or inactive, toxic or non-toxic, mutagenic or non-mutagenic. carcinogenic or non-carcinogenic. We simply use the binary terms to distinguish. Here, the concentration is not known, but simply we categorize based on the concentration values, of course. Now, let me take you to the statistical parameters in QSAR modeling. So, after QSAR modeling, how to validate, like whether the model is perfect or not, to check whether the efficiency of the model, we can measure through method of least square fit. That is actually we measure through R square value. And similarly, cross validation we measure through Q square and adjusted R square. Chi square test we can use. Root mean square we can use for checking the efficiency of the model. The model, whatever I mentioned, is the training data set model. I told you, no, there are two important terms. One is training data set and test data set. The training data set is actually you have known activities, known properties. But test data set we have to predict from the training data set. Okay, we also have bootstrapping and scrambling to produce a model more effective. Now, related to types of molecular descriptors, actually, I have circulated one journal paper called Scientific Reports from 2026, where my student has used machine learning technique. The machine learning technique, however, she has used is more related to QSAR kind of a relation. Okay, so that also takes all these properties, and if one or two properties we take in major consideration and we predict. Okay, so that's a kind of a machine learning model she has developed actually. Now let me take you to this types of molecular descriptors in QSAR. Actually, we have two types of descriptors. One is experimental measurements or experimental descriptors, and the other is the theoretical one. Experimentally, one can able to determine the log partition coefficient. How can we can calculate the log partition for a compound? Anyone know how to determine the partition coefficient, that is log P, for a drug through experimental approach? Because there are so many unknown terms. If you can make understandable to the others, you can speak. Can I mention you then how to determine the log P for a drug or a compound? For this, we use one concept, a chemical that is in, if you have the glasswares, even through the glasswares, we can able to predict. That is, instead of checking the partition coefficient in humans or animals, we can check even through the chemical experiments. The experiment what we use in chemical is to determine the octanol water partition coefficient. Octanol water partition coefficient. That determines you the log P. So whatever I told, octanol water partition coefficient, which is used to determine the log P of a compound, what is the methodology used here? Why we have to use these chemicals, octanol and water? Why can't we use some other chemicals to determine log P? Anyone have the answer for this? I just given the clue how the log P is calculated only through chemical experiment called determining octanol water. Why we are using these two chemicals? Why not other? And if you don't know, I can explain you. The octanol is having the, yeah, someone is answering, right? If you
Sir,
can
octanol has the maximum lipophilicity and it can the molecule can go into it. Water has this other ways.
exactly, ma'am. Your answer is right. And more accurately, I can able to mention that pH of octanol is 7.4 and pH of water is near to 7. Okay, it's near to 7, can be 6.9 based on acidic nature. Okay, approximately 7. So it means that the pH variation. Why we have to take a pH 7.4? Why can't we take some other pH? Any idea on this? For this, I can give you an explanation that the pH,
Sir, is it a neutral pH? Near neutral pH?
not exactly. The answer is actually Very good interaction. I appreciate. This is what I want, so that I can teach you more. Now let me take you to the octanol. The pH I told you 7.4. The major tissue present in our body is blood. No doubt in that. We have around 5 to 6 liters. So whatever drug we consume, we already discussed something called ADME. So during the absorption process, okay, it's going to happen only through blood. Okay, and distribution is going to distribute to the exact tissues where we want to target, isn't it? So if the absorption is happening in the blood, okay, so the pH of blood is 7.4. And you know that much of the component present in blood is nothing but water. We know more than 70% of our body is comprising of water molecules. Similarly, even in blood. So in blood, we have the lipophilic kind of a region that comes under pH 7.4. In hemoglobin, we have complex iron molecule in the form of porphyrin structure. It means that a heme, whatever mentioned, is nothing but a complex iron molecule. So this complex iron molecule related kind of a solid part can contribute to this kind of a pH, and the water part is going to contribute approximately to 7. And what is the purpose why we are calculating the log P through the chemical experiment? There is something called blood-brain barrier, BBB, blood-brain barrier. You know, the blood, I told you the pH is this. There is something called brain. The brain is more lipophilic in nature. Okay, but blood is we come because it comes under 7 to 7.4. Okay, so it is actually we consider as hydrophilic. The blood is hydrophilic and the brain is more lipophilic. The cranial membrane, whatever you find in our skull, okay, can find this is so. If you find that kind of is a more lipophilic, that is brain is more lipophilic. Whatever drugs we consume, if it targets the brain, okay, then it can lead to any kind of central nervous disorders or peripheral nervous system disorders. So, for most of the drugs are designed to target only the other parts, okay, other than the brain, isn't it? Like for a kidney, lung, liver. So all these things have following this blood. Okay, that is we have to target uh, through only the hydrophilic drugs, but only for the nervous system disorders, we have to target the brain. Even if you take paracetamol, you remember I told you an example when I just taken the smiles format of the acetaminophen and I just pasted in Swiss ADME, I can able to see from the boiled egg diagram, this acetaminophen is lying in the yellow color part of the boiled egg. It means that this is lipophilic. But generally for fever, there is no need to target the brain, isn't it? Unless if a person is suffering from psychotic disorders, okay, then only we try to provide these kind of lipophilic drugs. But acetaminophen is coming under lipophilic. Do you have any justification? Acetaminophen, if it is treated in less dosage, it targets only the blood because in the blood only we have an important target called COX-2, cyclooxygenase 2. We have to target that. But the same acetaminophen, if it is taken in excess, that is taking 3 or 4 doses at the same time, it targets up to the brain. I told you, even the patients can come to blinded state. They will be getting blind. And even if they take one more extra dose, it can lead to the coma. It means that it's targeting up to the central nervous system. Like this, it can check, okay, whether the drugs is a lipophilic or hydrophilic. Like, however, I mentioned for the log P, there are so many terms which we can able to determine even through chemical experiments. Even molar effectivity, generally we measure for the food samples, okay, can also be measured through the chemical experiments. The dipole moment, okay, polarizability, all this. And theoretically, whatever I mentioned, like a blood-brain barrier, all these things comes under theoretical. But experimentally, we measure through octanol water partition coefficient, and finally we determine log P value from this. So this is actually an experimental part, and this is actually a theoretical part, the blood-brain barrier. Okay, now let me take you to the classes of molecular descriptors. So if you see the classes, here I mentioned one is the 5 types of descriptors, but we also have 5D and 6D, which we'll be discussing in subsequent slides. Now first about 0D. The descriptors start from 0D. Constitutional descriptors or count descriptors. For example, if you take acetaminophen, how many atoms are present? Okay, simply if you find like that, that is 0D. 1D, we also find how many structural fragments are there, how many fingerprints are there. Okay, this is 1D. 2D, graphical variants. There is something like a 2D structure. However, you find a 2D structure of a compound, such a kind of a graph in variance. And 3D descriptors, we find 3D Morse descriptors, film descriptors, so many types of descriptors are available, including size, steric, surface, and volume descriptors. 4D is generally if you pack a compound in a grid, okay, then what are the types of descriptors? Even X, Y, Z directions you can able to find for that. So that is coming in a grid of COMFA methods, wall surf. Now let me discuss about the other dimensions. Okay, so other than the 4D, which is majorly mentioning about the conformation, orientation, and protonation state of the compound, in 5D, we can also check the induced fit parameters. Whether a ligand can talk with a receptor, the receptor, I mean a pseudo receptor, whether it can able to talk with a pseudo receptor or not. So such a kind of a docking kind of a thing, whatever a ligand we have, based on the functional groups, whether it's going to bind with the target or not, that kind of a prediction comes under 5D. And 6D, you can able to see the 5D QSAR combined with the solvation condition. For example, finding the binding sites, whether it's going to bind with the binding site 1 or 2 or 3 for that pseudo receptor. That also we can predict through 6D, 6 dimensions. 7D QSAR, real target based receptor model data derived. So this is the ultimate one. Okay, whether the compounds can bind perfectly or not. Okay, the finalization we can find in this 7D. ChemTest is one of the tools which we can able to use for prediction of QSAR. Okay, so you can find this information even if you visit ChemTest tool. Okay, we start with molecules and we use the fingerprints and we use it for many applications. Now this is an important slide. Now we can focus on this. This is a pipeline, how we work with QSAR. So in QSAR, I told you we have different dimensions from 0D to 6D, 7D. So whatever dimensions we have, if you see the 0D, we simply have the molecular formula. In 1D, we have this kind of a structure or just a smiles format, whatever we have. In 2D, a 2D structure. 3D, you find the 3D structure. 4D, you can also see the spatial conformations. What is the best ligand position with respect to the functional groups? Okay, like this. So for this, the molecular descriptors, what we find is constitutional, electronic, geometrical, hydrophobic descriptor, lipophilic descriptor, steric, quantum chemical, and topological. These are the molecular descriptors. So now, if you know the type of dimension of QSAR, then if you know, you have to select a few molecular descriptors, and based on the selection of molecular descriptors, one can go with multivariate analysis. So these are the methods, machine learning algorithms we can use to determine the activity of test data set either through regression method or through classification method. So these are the methods we use to determine the activity of unknown compounds or unknown activity of compounds. Okay. So this is actually a kind of a graph that is plotting experimental activity versus predicted activity. So from this, one can able to draw a kind of a line, a linear line, and you can able to find what is y value, what is x value. Or from that, if you know y and x, then we determine m and c from that. And finally, model validation can be done through this. Now, about software used in molecular descriptor calculation. E-Dragon, Codesa, so all these things. Actually, this is according to ChemDes, you can ignore. Now, like however we discussed about QSAR, the general term what we use in other terms, is a property. If property is activity, then it is QSAR. If property is related to retention, where we can find this retention? This retention we find in chromatography. You remember in chromatography, we can able to separate the compounds based on the color, based on chroma, that is color. So each and every compound is going to separate on with respect to the time, isn't it? Myself, I worked on withanolides. Withanolides or steroidal lactones present in ashwagandha. If you see withanolides, we have around 41 at present. Now even they are increasing. Okay, based on recent discoveries, they are still increasing the number of withanolides in ashwagandha. So if you have 41 withanolides, if you see the structure is more or less same. Molecular weight and other kind of properties are very same. Then even if you want to separate, our company is focusing on even selling the product from ashwagandha, especially withenoids. We are selling around 7 products, 7 different withenoids from ashwagandha. Each and every withenoid, like vithaferin A, withenoid A. So these are going to separated based on the retention times, based on the mobile phase, whatever we use, and the time of run, we can able to see the vithaferin A is separated in one particular time, and withenoid A is going to separate it in another particular time. That's what we call it as retention time. And even if you have compounds which is very closer, even that can be that kind of an retention relationship we can study through QSRs and QS chromatography relationships. Again, the same kind of application you can find. And T stands for toxicity. My my student has worked on QSTR. Okay, QSTR in the scientific reports recently published paper. One can look through that, how she has predicted the toxicity. She has used the Protox tool and predicted the toxicity and used machine learning techniques to predict for the unknown compounds. Now, let me take to the quantitative structure electrochemistry relationships. That is the chemistry persons. Okay, for electroplating, they use this kind of an electrochemistry relationships, biodegradability, like the environmental engineering engineers generally use this biodegradability studies. Okay, this is just a journal paper. We have one kind of a journal like a journal of liquid chromatography. Liquid chromatography, gas chromatography. These are the general chromatography techniques we use. Okay, and QSAR. What is QSAR equation? I told you from the beginning. So whatever we predict ultimately at the final is the activity. This is nothing but function of physico-chemical properties or structural properties, including with the error or bias. This can give you the accurate kind of a correlation. So one can able to find this is to just develop a mathematical relationship. How we use the MATLAB to determine the correlation. Similarly, we can use the QSAR for it. 3D QSAR we can able to predict from these well-known tools like COMFA. It means that comparative molecular field analysis. This is present in one kind of a tool called Cybal. Okay, Cybal is a product of Tripos, and this Cybal has modules like COMFA, COMSIA for predicting through 3D QSAR. So this is a workflow for predictive QSAR model. So if you see this, I told you already, we have a training data set and we have a test data set. So test data set can be predicted from the training data set. So more details you can find in the pipelines. Let me explain further in the subsequent slides. This is also kind of a pipeline. like taking a data from electronic databases, literature data, lab collections. We store in the data collection, then through the data repository, we predict the QSAR through QSAR models. Plenty of regression and classification submodels are there. Use that models and you predict it. And this you can use it for virtual screening. Like if you have 100 compounds, if you want to find which is the best 10 compounds, you can able to use the QSAR models. So after screening to limited number, then we can go with the experimental validation. QSAR protocol. Start with a curated chemical data set. That's what I call it as training data set. For curating a chemical data set, we have this much sub-steps: removal of inorganic compound, structural conversion, normalization, treatment of protomeric. So you can find this. Next, we have to proceed to the stage 2, descriptor generation. Then split into training and data. Training, we can increase it 80% or more than that, and the test data set we can go to 10 to 20% of the total data whatever we have. Even like this also we can split into 2 different files. Okay, so even in through the Python programming, if recently I have conducted a course. I mean, last month I conducted a course that I explained you how to do the QSAR through Python program. Today, I'll be explaining you to how to do the QSAR through Excel. This is a very simplest method one can able to use. Okay, so I'll be teaching you how to use the QSAR through the Excel. So building QSAR, validation of the QSAR, virtual design. So these are the sub-steps and subsequent stages. Now, let me take you practically something about 3D QSAR. So 3D QSAR through COMFA methodology. So from the data set collection, first I suggest you to determine, to first create a kind of a data set from 20 compounds or more than 20 compounds. And whatever data set you have created, then you go with the molecular structure preparation. Because COMFA is nothing but 3D QSAR. Okay, then we go with the pharmacophore alignment. What is pharmacophore alignment? That will be discussing. Like we have a hydrogen bond acceptor, donor, hydrophobic aromatic regions on the surface of the lead compound. This we can use it for determining the structural affinity. Okay, more about this we'll be discussing in the subsequent slides. And placing the ligand in the 3D grid, and then field calculations like a static field and electrostatic field. Then partial least squares regression analysis. Then we go with the validation to determining Q square, R square values, root mean square, error, etc. Then we go with COMFA contour maps. Like for the ligands, we map the regions where it's going to bind with the receptors, which is the most important functional groups, which is going to bind with the target receptors for disease, for disease state. Okay, that will be mapped here in contour maps of COMFA. Then we come up with a kind of a lead optimization. Initially, we have a truck like this, but after QSAR and after the lead optimization followed by the QSAR, you can find an analog is developed. This structure is different, but this structure is more resembling to that, but a slight different structure has formed just because a previous step we have used contour mapping, which is equivalent to pharmacophore mapping. In pharmacophore, we have pharmacophore alignment is different, pharmacophore mapping is different. So pharmacophore mapping is coming under hybrid-based drug design. But this comes under ligand-based drug design, pharmacophore alignment. So after lead optimization, we can apply in drug design applications. Okay, that is what virtual screening. Similarly, we have another 3D QSAR through COMSIA methodology. Again, this kind of a tool is present in Cybele. Like however we have Discovery Studio, we also have Cybele, another kind of a software, where we have these kind of a modules to practice with 3D QSAR. Almost more or less a similar kind of a protocol we have to use. Here now I'm going to distinguish about COMFA versus COMSIA. Comparative molecular field analysis, here it is molecular similarity indices analysis. What is the difference between these two? If you see, if you see the probes, whatever we use, this is sp3 carbon, but here we have sp3 carbon electrostatic interactions like positive or negative ions in the atom, hydrophobic interactions, hydrogen bond donors, hydrogen bond acceptors, everything, all these things are collectively considered as multiple probes. We have a single probe, but here we have multiple probes. That's the major difference. In similarity indices analysis, we instead of simply restricting up to the steric field and electrostatic field, we also focus on hydrophobic, hydrogen bond donor, hydrogen bond acceptor. So we find detailed kind of analysis in COMSI. Got it? This is the major difference between these two packages, these two tools present in Tibel package. So this is more reliable when compared to COMSI. But we need more data, more comprehensive fields. The data set, whatever you prepare, is time consuming. But if you have a simple data, we can predict it as soon as possible using COMFA. So these are the advantages and disadvantages of these two methods. And there is one more tool called Galaxy. This is used for predicting QSAR. This is an online tool. We have an option called for building QSAR model here. And after that, we can predict activity. So through these kind of links available. So whatever QSAR, using the QSAR, we have developed the linear regression model. To the linear regression model, we have predicted the equation y equals mx plus c and r square value. Here, the data is whatever you see or nothing but drugs, like drug 1, drug 2, drug 3. If you have this much number of drugs in a data set, and if that is mapped versus log IC50 experimental versus log IC50 predicted, then you find these kind of dots lying in this. We simply have to come up with a kind of a equation, with a kind of a line with an equation which can show this r square value also. If the r square value is close to 0.99, then the model is more accurate. If r square is up to 0.95, it's okay, we can use it. Okay, still I can tell we can use it, but this is more accurate. But if you go beyond, less than 0.95, that is not preferred. Okay? The model is not that much correct. If you go 0.90, obviously we can decline using the model. Then how to increase the R square value? Instead of using small number of drugs, we can increase the size of drugs. Instead of 15 drugs, if you go with 150 drugs or 1500 drugs, R square value definitely is going to increase. Okay, increase the size of the training data set to increase the R square value, which provides you the quality of the model. Okay, now let me first explain you about how to practice this QSCR. What is the time? Okay, still we have 10 more minutes. Now first I take you to the Excel. Am I audible to everyone? Yes?
Yes, sir.
Or is that? Thank you. Now first I will be explaining you like how to predict a QSCR through a simple Excel. So whatever Excel I have, I just have these properties. See the properties. So I'm going to have nearly 20 compounds. For example, for virtual screening using Autodoc PVRX, when I searched through a keyword called pyrivinone, I can able to find around 15 pyrivinones out there. Like pyrivinone A, B, C, D, E, F, G, H. So 15 compounds out there. So you take this kind of an Excel, okay, have all these compounds like 1 to 15 compounds, and if we want, we can also fit the smiles format. And whatever I have docked, even if you take all the 15 compounds and if you dock with one particular protein target, you get the IC50 values. So just input all the IC50 values, whatever we generated from PVRX. Then whatever here I have given the values are nothing but docking energy. You know that even autodoc PVRX can give you energies, negative maximum energies. If you dock a peptide with protein, it can also give positive energies. Peptide gives a different kind of energies, but compound gives different energy. The energy, whatever we get a binding energy docking with a compound, should be less than minus 6 kilocalorie per mole. Then only can tell it is more significant. For example, if I just gave these two, if I filled only two properties alone, but you can also try with other things also, like hydrogen bond donor, acceptor. If you want to do take hydrogen bond donors and acceptors, no need to even do the docking. Simply go to the PubChem, and for that 15 compounds, you just to go to that particular compound and fill these values alone. You can also get TPSA, total polar surface area, rotable bonds, anything like that. And including the molecular weight, log P, everything is there in PubChem itself. No need to go with any other tools. Only thing is that you have to go to the database, and for the 15 compounds, you have to fill these values. Now at present, at randomly I filled two columns. So with these two columns, now I'm going to explain you how to predict a model equation through linear regression method that is equivalent to preparing a QSAR equation. Okay, so for this, first fill these 20 compounds values, select the 20. Now click the control and again select another 20. Now if you see these two columns are selected with the 20 values, 20 and 20, 40 values. Next thing is to go to the insert. Inside the insert, you can go to the recommended charts. If you click the recommended charts, default it's giving the scatter plot. This is the base plot, a recommended plot. Simply click OK. So now we can able to see this docking that is activity. I mean docking energy versus activity. Docking energy is in y-axis, and whatever activity values I typed is in x-axis. So whatever 20 ducks values I have filled, this is present in this now in the scatter plot. Now, how to determine a QSR equation? For this, simply select. So if you select, all the dots are got selected. Okay. So simply if you select it, but few dots are not selected. Let me check whether I can able to select all the dots. Still control. So this is the maximum number of dots I can able to select if I click once. Now the next thing is you have to click plus, go to the trend line, or else I can, after selecting this, I right click, add trend line. You find this add trend line, click this add trend line. So if you see this, we can able to see it is a linear kind of a regression. Okay, we also have logarithmic and polynomial, etc. So I'm going to click something like display equation on chart, display R square. Okay, if you click this set intercept, then it default take like 0 value, but we want even the C value, so I'm not going to check this. Only display equation, display R square value. See, the equation is predicted. Whatever equation we have predicted, okay, the R square value is what actually 0.12. It's very weak, weakest model. The R square value should be close to 1, but we got 0.12. It's a very weak model, no doubt in that. And you see, only these two drugs alone is joining this line. This is according to linear regression. But now I'm going to select once again all these points, and now I'm going to try with another kind of a model for this add trend line. So instead of linear, I'm going to try with the logarithm. So even log p partition coefficient, if you have a partition coefficient kind of equation, the logarithmic can give a more accurate results. So I try with a logarithmic kind of a regression model. Again, I'm going to check this display equation, display R square. So this is what the equation for the uh, logarithmic thing, whatever I predicted. Now you can check this. You can find the different x values. I mean, you find 2.27, here it is 0.29. So the intercept is different. Now R square, you can see it is less. When compared to linear regression, the logarithmic regression is not good. So this kind of inference you can infer from this. The R square should be close to 1. So logarithmic regression is not good model when compared to linear regression. So this is how we can able to determine the kind of efficiency of the model through Excel, the simplest method. Even if you go to the QSAR kind of a tools like a COMFOR, COMSA, okay, we start with initially the 1D and complete up to the 3D structure. Unless you have a 3D structure of compounds, we can't able to do. After creating the 3D structure of the compounds, then we can go with these kind of methods. Got it? So these are the steps we generally follow. Okay, if this is something about QSAR, now let me take you to the next thing, that is pharmacophore modeling. You know, the pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. It looks like a docking, isn't it? You know, I told you in pharmacophore modeling, we have a normal pharmacophore alignment and pharmacophore based docking. That is pharmacophore mapping and pharmacophore based docking. Okay, the docking is what hybrid based. That is, you should dock a ligand with a receptor to find whether the ligand is binding perfectly or not. If you see in docking, we measure through binding energy, but whereas in pharmacophore, we measure through fitness value. Instead of binding energy, we give through fitness scores. Okay. Now let me explain further in the next slide about pharmacophore model. See, you have one kind of a drug, and for the drug, we have so many kind of functional groups. Each and every functional groups, okay, it can be a hydrophobic, hydrophilic, lipophilic, steric. So all these things are shown in the spherical kind. So this sphere can bind with one part of a receptor, and this sphere can bind with another part of the receptor. So like this, we can distinguish. But what is the most important functional group in the ligand which binds with the active site? That's most important, isn't it? So before talking and after lead generation, we go with this method called pharmacophore modeling. That is before talking and after lead generation. So if you see the features of pharmacophore, I need three to four more minutes to complete. So if you see the features of pharmacophore, we can able to see the ligand receptor interactions are polar positive, polar negative, or hydrophobic like this. More about this, I'll be explaining in the subsequent slides. So this is similar to QSAR. We have training data set and we have test data set. The test data set can be predicted from the training data set. Steps in model development. Now, I directly take you to the kind of an experiment how to work. To work with the pharmacophore mapping, we have a tool called Form Mapper. In this, we have to simply upload a ligand structure. In this case, I have uploaded goirivinone B in STF format, whatever I downloaded in PubChem. After uploading this kind of a chemical compound, I got these kind of results. You know, from these results, we can able to see that the ligand is perfectly binding with this PDB ID. So how I am telling, so through the fitness score, the maximum score we got is 3. Like for example, if you go to the abroad and go with the master studies, we have the maximum of 4 CGPA scale, isn't it? So one can able to see however ranking up to the 4 CGPA. Similarly, the fitness score, the maximum what we get from the FarmMapper is 3, and this is ranked in descending order based on the fitting of the ligand with the receptor. So you can able to see the pyrimidine B is next to talk perfectly with this DNA-directed RNA polymerase. Actually, the RNA viruses have these kind of protein. So this pyrimidine B can also use as an antiviral drug. Similarly, can find other target receptors. This looks similar to the target deconvolution approach. But in target deconvolution approach, we measure through binding energy. But here we measure through fitness score. Okay, one maximum score, and progressively you can see descending of scores and the corresponding receptors. If you click the first link, you will be getting like this. A ligand and the corresponding pharmacophore functional groups are shown. So whatever shown in this blue color are most important. Next to that is the pink color groups. Because the pink color stands for the acceptor, whereas the blue color stands for the hydrophobic interactions. These undergo the hydrophobic interactions, and these undergo the acceptor kind of an interaction with the target receptor. Got it? Form mapper. That's it. So now, related to form mapper, I just go to the website and show you how to make it, how to practically work with it. Because tomorrow we go entirely with a different concept called vaccine design. Form mapper. So if you use this form mapper or even the link, whatever I typed here, oh, copy, sir, I typed something different. Okay. No, I take it to the form mapper. Okay. Form mapper. Let me take it to the server. Yes, this is the URL. I just type in the chat box. So you have to click this. After going to this, simply click submit job. After clicking this submit job, you have to upload the STF format of the compound. For this, actually we already downloaded an STF format, I believe. If you go to the desktop, okay, see a DVD where I exactly stored in STF. I think acetaminophen is in STF format at present. Yes. So I'm going to upload this acetaminophen. Later, I can give email address if the time taken to receive the result is late. If I can't able to show you the result today, tomorrow I'll be showing you the result because I will be receiving the output in my email ID. Simply click continue. Just upload one compound in STF. Type your email ID and give the description what exactly you are searching. Don't submit more than 10 jobs. First, for example, if you're trying, you try like this. It takes 1 or 2 minutes to generate the results, I believe. If I have already kind of form mapper, I can try to open. Let me check whether I have. It is in the repository or not. Still, it's running here, but I'm running one more thing which is already executed. So that kind of a result is similar to however I showed you in the PPT. Okay. Okay, I have completed my session. Anyhow, tomorrow, after getting the result, I show you. Any doubts you can ask me. I have completed today's session. I have completed QSAR, how to practice a QSAR using Excel, and how to practice pharmacophore mapping using form mapper. Any doubts?
Sir,
Yeah,
sir,
please.
is this QSR tools are freeware, sir?
Which one, madam? QSAR, are you asking? In
Sir, name.
QS, in
Yes, sir.
QSAR, COMFA, COMSA, and whatever tools I projected in the PowerPoint, all are commercial tools. In
Okay, sir.
QSAR, we don't have freewares. But Galaxy QSAR, I told you, that actually is just a model. Like if you want to provide a demo to the students, okay, you can use that website. But for practical research, this is not sufficient. Okay, we
Okay, sir.
have, yeah. So whatever data set we have to practice, then we should have the commercial tools.
Thank you, sir.
Yeah, thank you. And any other participants have any other doubts, you can ask me. If no doubts, thank you very much for your patience and interaction from the beginning. Have a nice day. Good night to everyone.
Thank you, sir.